Hi, my name is Amin. I'm a graduate student of neuroscience at Columbia University, and today I'm going to present my accepted poster in COSYNE 2021 titled Predicting Perturbation Effects from Resting State Activity Using Functional Causal Flow. To motivate this, our goal is to develop a causal metric and have a reasonable guess about where to stimulate, basically to predict what would happen in the perturbed state of a network only by looking at the resting state data. This is important experimentally, both because of designing effective brain-machine interfaces and also being able to predict the perturbation sites in the brain disorder treatment, and also in designing microstimulation experiments for causal scientific investigation. Theoretically, it's an interesting problem because identifying the causal interactions in a network of neurons um, is only by looking at the resting state data is a challenging mathematical problem, and also because current directional connectivity metrics perform poorly. So there was a challenge called the first connectomics challenge, in which the participants were asked to only use the resting state time series data and recover the connectivity matrix of the nodes in this network. And as you can see in this results table, which I took from the paper, there's a dramatic decrease of the accuracy when you go from the undirected network to the directed network. Um, so what we did is that we used the resting state data and developed a metric which we call functional causal flow, or FCF for short, uh, that predicts what is the perturbation effects in the stimulated state. And we applied our framework to rate networks, biologically realistic spiking networks, as well as real data from monkey brains. To describe the method, I'm going to first explain this theorem, which is called Tekken's theorem. So this theorem states that if we only observe one single signal from a multidimensional dynamical system, so let's say that this x1 to n t is a multidimensional dynamical system, but we only get to observe xi of t. If we build a delay vector, so if we grab the signal at time point t minus tau, all the way to t minus d tau plus tau, basically if we shift the signal multiple times, and so these are the shifted versions of the same signal, this gives us a three-dimensional vector in each time step, and then we can plot this three-dimensional signal, and the theorem states that this manifold that we recover here is in one-to-one relationship with the original data generating dynamical system. So we can use this framework, and given that we have two different signals, we know that if they are parts of the same dynamical system, if xi and xj are members of the same dynamical system, then these two maps, one-to-one maps, exist between xi and the data generating dynamical system, and also xj and data generating dynamical system. So if these are members of the same dynamical system, by combining these two maps in this way, in the following way, we can construct another map which can transform the delay vectors of node I to node J, and this motivates us to develop our metric, which is the following. So the algorithm that we use based on this principle is that for every pair of signals xi and xj, we first create their delay vectors and then divide them into train and test, and we infer this psi ij based on the training set and using nearest neighbors method, and we apply it to the test portion and use the reconstruction accuracy as a surrogate of the causality. So we know that if xi and xj are parts of the same dynamical system, this psi ij can perfectly reconstruct the test portion of the data, but if they're not in the same dynamical system, then this value will be a low value. So to test this framework on the synthetic data, we first generated a network of upstream and downstream neurons. So we have two subnetworks in this simulation, and the blue neurons are unidirectionally connected to the pink neurons, and And then we can, for every pair of neurons, we can compute this metric, the FCF metric, and this gives us a matrix of functional causal flow values. And as we can see here, this metric can recover this sort of hierarchical structure in this network, and it can predict that there's a causal flow between neurons 1 to 3 and the rest of the networks here, but not between the downstream neurons to the upstream neurons. We can so this is interesting because we can kind of recover this hierarchy in the network, but the more interesting thing for us, which is the main motivation, is to predict what is the perturbation effects. In order to have a better definition of what do we mean by the perturbation effects, we are going to define this notion of interventional connectivity. The way that we define it is that we actually apply the perturbations in the network. So for every pair of neurons I and J, we stimulate neuron I, say that we are stimulating neuron 1, and we measure the activity in neuron J before and after stimulation. If neuron I is the upstream of neuron J, if we perturb neuron I, neuron J will actually change its firing rate. So this is the principle that we use to define this metric. We look at the distribution shift of the neuron J before and after the stimulation of neuron I, and that can give us another matrix, which we call the interventional connectivity matrix. So this matrix tells us for row I and column J, if I perturb row I, what is the effect of that perturbation on column J? And interestingly, we observed that the elements of this interventional connectivity matrix are correlated with the elements of the FCF matrix that I described earlier. To further test this hypothesis, we generated data from a clustered network of inhibitory and excitatory spiking neurons, we showed that the FCF can not only recover the clustered structure of the network, but also the FCF values are correlated with the interventional connectivity metric that I just described. Finally, we tested our framework on the real monkey brains. So we have resting state recordings of the monkey brain in the prefrontal cortex, and we also deliver stimulations to individual channels and record from all the other channels. And again, we can compute the resting state FCF in the top row, and the perturbation or the interventional connectivity in the bottom row. Every column here is a separate channel. And again, we can show that the FCF is correlated with the interventional connectivity. So in summary, we developed a metric to infer a causal hierarchy in the network of interacting nodes, and we tested our framework on synthetic and real data from the monkey brains. And also we developed a code package for doing all these types of analysis. So our code package has multiple classes for causality, for computing FCF and interventional connectivity, as well as performing delay embedding principles, also running multiple simulations from networks that are used in neuroscience experiments. And the codes are pretty clean. I encourage you to take a look at our GitHub repository. And as future directions in terms of the applications, it is interesting to apply this method on other data sets, calcium recordings or fMRI or wide field imaging. And theoretically, it is interesting to investigate this relationship between the resting state and the FCF and the perturbed state and this interventional connectivity, to gain insight of why these two metrics are correlated. I would like to thank my collaborators and my funding sources and my advisor, as well as you for listening.